What is a MUSE stem cell treatment? In this video, we're going to talk about that there's no single IV infusion, no surgery, no immunosuppressants, no tissue matching requirements, and 68% of stroke patients reached functional independence at 1 year. And that is what a MUSE cell treatment actually looks like in the clinical trial data. And it does not resemble anything else currently available in regenerative medicine. I'm really excited about these MUSE cells. And in this video, we'll uncover what they are, where they come from, how they were discovered. So stick around. I'm Regan Archibald, for those of us who haven't met, founder of Ageless Future. We're the world leader in longevity medicine. We want to bring you the most innovative updates when it comes to your longevity and your future. And so we run blood labs and get the right testing on every single person we work with. For those of you who want a team of providers who care about you, who give you the right lifestyle and the right interventions, all you need to do is go to agelessfuture.com. Let's talk about MUSE cells. So the clinical stage MUSE cells, there's a product called CL2020, and it's an allogeneic, meaning it's a donor-derived, not your own, cell preparation manufactured by Life Science Institute in Japan under the pharmaceutical GMP protocol. So allogenic means off the shelf. These cells are manufactured in advance, quality control. They do not require taking anything from the patient. And the administration is of a single intravenous infusion of 1.5 to the times 10 to the 7th power of donor MUSE cells. Now, 15 million cells in 15 milliliters given over 10 to 15 minutes. That's a lot of 15s and 10s. So no HLA matching, no immunosuppressant drugs before, during, or after. Now, that's pretty interesting because MUSE cells, they don't contain these human leukocyte antigens that your immune system would look at as being a threat. Every other allogenic cell therapy on record requires immune suppression to prevent any kind of rejection, but MUSE cells escape this requirement through a placenta-like immune tolerance profile. They co-express this HLA-E, this FAS-L, and PD-L1 surface molecules that signal immune neutrality. So it's like, hey, we're just friends, we're here to help. And that same mechanism that produces a genetically distinct fetus from maternal rejection appears to protect the donor MUSE cells from host immune attack. So 2 trials worth knowing. First, the Journal of Cerebral Blood Flow and Metabolism 2023. They took a randomized placebo-controlled trial in subacute ischemic stroke. And this is where they had 43 enrolled, 35 completed, 25 did the CL2020, 10 did placebo. So out of the 35, 10 had placebo, 25 did the MUSE cells. And a single IV infusion was given 14 to 28 days post-stroke. At week 12, 40% of the CL2020 or the MUSE cell patients reached a modified Rankin scale score of 2 or below, meaning functional independence. That's phenomenal after a stroke, versus in the placebo group, only 10% reached that score of 2 or below. So only 10% versus the MUSE cells. So the preset efficacy threshold was exceeded at week 52, and 68% of the patients had reached another level that significantly shifted towards a lower disability score across the treated group. This trial was done in Japan. Yamashita was the author. Another study, Cell Transplantation 2023, they looked at a phase 2 ALS trial. So ALS is a brutal condition, the amyotrophic lateral sclerosis. It's a progressive neurodegenerative disease, something that I don't wish on any of you or anyone. What they did is a small cohort, 5 patients, open label, repeated IV infusions of 15 million MUSE cells per dose once monthly for 6 doses over 12 months. There was no drug-related serious adverse events across the full 12-month observation. They also found the functional progression scale for ALS showed stabilization trends in 3 of the 5 patients. Now, I know this is small, but we're learning about what this can do, and I can tell you there's not a lot of great treatments, if any, for ALS. They also found the serum S1P, the homing signal, released by dying cells declined progressively over the treatment course, consisting with the cells responding to helping resolve the injury signal. Now, these are small trials, but once again, there's going to be more in the future. So what the hype gets wrong and what it gets right. It was not attributed. So a lot of people say, well, there was someone who had a stroke in one of the trials, but what they found is it was unrelated to the MUSE cells. And even people who've had strokes, these are medically fragile people. While MUSE or the CL 2020 is not FDA approved, it's not commercially available in the United States, we are seeing a very large amount of enthusiasm. And many people in the longevity and regenerative space, we've looked to the Japanese based on some of their core policies and principles of longevity and extending life extension, and they've done some phenomenal things with the cell therapy. But the beautiful thing about MUSE cells is they don't require any kind of immune matching. There's no immunosuppression required. This is something that in every regenerative space, especially with induced stem cells, when you take stem cells and then you take them and you feed these stem cells and you grow greater populations and then maybe genetically engineer these cells to take on a particular cell lineage, there will need to be some kind of immunosuppression in many cases. In the MUSE cells, they seem to work very favorable even when they come from placental or umbilical derived sources. So for those of you who are considering this, well, the very first thing you want to do is get your blood work ran. Make sure you're working with reputable places in using MUSE cells. Once again, there's nothing that's FDA approved in the United States, but there are some very interesting things that you can start doing right away to activate your own MUSE cells. And so for those of you who would like to learn more, all you need to do is go to agelessfuture.com, book your free intro session, meet with our team, and then get your blood labs ran. We'll help you meet with one of our regenerative providers to find out what we can do in the regenerative medicine world for you. I'm Regan Archibald, appreciate you being on the channel. And for those of you who are learning more about MUSE cells, I'd love to hear what you've come across and any studies that would be of interest. See you in the next video.